Okay, so we have been discussing about the life cycle or the lifespan of a cell, in other words, cell cycle, and we have discussed in the previous class on Cisco how the entire life event or the time from where the cell is conceptualized till it further may divide into two daughter cells, or its life cycle or the cell cycle can be divided into the interphase, which is the longest phase of the cycle, and then the short-lived mitotic and cytokinetic phases. Of course, the interphase or the I phase. So inter means intermittent. So it is the phase in between two mitotic cycles or division cycles of the cell. It is further subdivided into GAP1 or G1, S or the synthesis. In some books, you will find it written as R or the replication phase, and G2 or the gap phase. So G1 and G2 are essentially GAP1 and GAP2 subphases meant for the production of proteins and duplication of cell organelles and enlargement of the cell size and dimensions, basically preparations for the division. It is the S phase or the replication phase or the synthesis phase, which involves the replication of the chromosomes, right? So basically the DNA content in the chromosomes, it doubles up. So the DNA, the total genome, the total nucleic acid content, DNA content doubles. So if the initial amount of DNA in the cell was 2C, during S phase, it will become 4C, right? Please remember this cardinal rule, which is the number of chromosomes does not double. It is the chromosome content in the chromosomes which doubles. So please ensure that, right? And of course, to recall this concept, remember I have already shared with you the structure of a replicated chromosome should bring you back. So you can pause the video and you can, ensure that you memorize these various sub phases of the interphase and you remember what all is happening. It is essential to note that during S phase, it is not just involving the duplication or the replication of the DNA content, but it also involves the duplication and synthesis of the centrosomes. Why centrosomes? Well, the centrosomes have the perpendicular centrioles. They are going to produce the microtubular rays, which are ultimately being called the spindle rays or the spindle fibers. And it is to the spindle that the chromosomes will attach. And this is very essential because the mitotic phase depends upon the duplication of the centrosome, right? But sometimes the cell may, decide that it has formed two daughter cells. Okay, but the daughter cells, either they may continue with G1 and the entire cell cycle again, or the daughter cells may exit the G1 phase and they, over here, somewhere around here, they will prefer to enter a stage called the G0 phase or the quiescent stage. So this is the resting stage. Now, why would certain cells take this rest or pause from division? Perhaps they are becoming differentiated or specialized to perform some specific roles or functions. So they are not looking forward to division, to active division. They are looking forward to differentiation. For example, neurons. Neurons are meant for conduction of nerve impulses, right? So they are differentiated. They are specialized for their role. They are no longer going to divide, right? So for some cells, it may be a permanent stage of rest. For others, some cells may have the ability to revert back to the actively dividing status quo in special circumstances. So your assignment is to tell me in the comments below, what are those cases of cells in which the reversion from the differentiated stage can be caused, which brings you to the term de-differentiation. So remember three terms: differentiation, de-differentiation, and re-differentiation. Looking forward to your answers in the comments below. Now, because the cell cycle is a very regulated event, right? So it means the cell is constantly checking for any errors or any inaccuracies. So it will halt itself at that stage, try to correct the problem, and then proceed the cell further in the cell cycle. If it is unable to do so, cell cycle may be halted or stopped irreversibly. So this brings us to the next topic, which is the cell cycle checkpoints. So checkpoint is that point in the cell cycle of a eukaryote when it assesses for any inaccuracies, tries to repair and correct them. If not possible, then the cell cycle will be halted. So you have three main or essential checkpoints. The first is the G1 checkpoint at which phase G1 of interphase. Then second is the G2 checkpoint, which is at the G2 subphase of interphase, and then during the M phase, before anaphase, before the chromosomes they segregate and start moving towards the opposite poles, we have the mitotic checkpoint at the M phase or the spindle checkpoint. And what is the specialization of each checkpoint? So I'm just pausing here for a moment because I want you to take down this definition of what is a checkpoint. So it's a regulatory stage at which the cell will internalize and externalize its signals, its cues, whether the cell cycle should proceed or should it be paused, corrections should be made. So whether you are to proceed with cell division or not, right? That is being assessed here. And then we have the first checkpoint, G1. So this is also called the first main decision point of the cell, where the cell will ultimately and decide whether to proceed with the division or not. So what are the four main factors the cells are at this time internalizing? Number one, the cell size. Is the cell large enough? Has the cell grown enough during G1? So should we proceed towards S, that is replication of the centrosomes and the DNA? So if the cell size is not right, this will be paused. Second, does the cell have enough nutrients to act as reserve food materials to energize the entire process of division, right? Then three, it checks for any molecular signals, any growth proteins or growth signals or factors required for the process of cell growth, are they there or not? And then does the DNA need any correction? So before the DNA, enters into the synthesis phase, it is proofread. It is proofread for any inaccuracies, right? So all of this is checked during the first checkpoint. And what do we have during the second checkpoint, which is at the G2 phase? So this is there to ensure that the cells are healthy, they are undamaged, the DNA is not damaged, the integrity is there. And so G2 is coming after S. So they are also going to cross-check if during the synthesis phase, the DNA was completely copied, were two complete copies of the parental DNA produced, because equal division among the daughter cells has to happen. Right? So all of these checkpoints are there. And then the last case, the third checkpoint, which is called the mitotic checkpoint or also the spindle checkpoint. This ensures, the cell ensures at this stage that the chromosomes, they are attached to the spindle fibers at the disc-like covering of the centromere called the kinetochore, right? And that the chromosomes are aligned at the spindle at the correct position. So the cell scans for any misplaced chromosome, any chromosome which is at a position in the cytoplasm where it was not supposed to be. Are the centromeres, are the kinetochores of the chromosomes nicely attached to at least two spindle fibers? Because once the cell will enter the anaphase, the chromosomes will be pulled apart and there is no looking back from it. And the entire cell cycle, in this case, these various checkpoints of regulation by the cell, this is supplemented by certain regulator proteins like cyclins, cyclin-dependent kinases, also called the CDKs, and the APCs, the anaphase promoting factors are also involved in this case. And of course, there is this diagram which tells you that how are these different regulatory proteins like the cyclins, the cyclin-dependent kinases, and the anaphase promoting factors and cyclins, how are they involved at these various checkpoints, right? So we will be proceeding with the complete detail of these regulatory proteins and mitotic poisons and spindle poisons like colchicine in the coming class. So today is more about, you know, internalizing the process of cell checkpoints, because this process is very essential for the cell, the division process. And because it's such a main event and so much of preparation is going on during interphase, it has to be ensured that there are no errors or inaccuracies. Otherwise, at either of these three main checkpoints, the cell will not proceed with the next stage. So the cell may not proceed to synthesis of the centrosomes and the DNA. The cell may not proceed to enter to enter into mitotic phase or the division phase. The cell, even if it has entered the mitotic phase, may decide to halt, pause before anaphase. So please remember that these checkpoints are being controlled by the cyclins, cyclin-dependent kinases, and the APCs. I'm going to talk more about these proteins in the coming classes. Of course, I have already discussed some essential questions to you. You may pause the video at different points, note down the details which are absolutely essential to you. And while you do that, I would also recommend going through the NCRT for the same. It's really essential for us to see what is there in the NCRT. So, of course, there's this diagram in which you are shown how the daughter cells may choose to exit G1 and enter G0 or the quiescent stage. Another thing is, I've already discussed in the previous classes, the duration of the cell cycle. Now, this is very variable among different organisms. Let's say a typical human cell in culture, when it is being cultured in vitro in lab conditions, takes 24 hours to complete one life cycle. So that's one duration. So you have yeast taking 90 minutes, for example, right? You have E. coli taking some 20 minutes, for example. So you can very easily calculate about 4 to 5% of this is the actual division time. It's the actual mitotic time and the cytokinesis time. So please ensure that you go through the NCRT. Again, the fact that I was mentioning to you about that has been stressed that the number of chromosomes will remain the same, but the DNA content will double. So 2C will become 4C. This concept has been reinforced in the NCRT and you must make an effort to go through that. The quiescent stage has been discussed well as well. And, you must understand that many a times the cells, whether they are the skin cells or they are the cells of the internal lining of our organs, constantly cells are being damaged. They are being lost because of cell death, natural aging or senescence, or due to disease, infection, pathogenic attacks, etc., or any kind of environmental stress. So it's very essential that the cells should have the ability to revert back to the stage of active division. The cells which have entered quiescent stage or G0 phase should have the ability to revert back in order to make up for the loss of cells due to injury and cell death, right? So this is very essential. Now I want to give you some terms over here for assignment before we talk of the M phase, the cell regulatory proteins, the checkpoint proteins. Before we talk of prophase, metaphase, anaphase, telophase in the coming classes, I want you to tell me in the comment section below the difference between programmed cell death, PCD or apoptosis, versus senescence or aging. And that's it. We'll be meeting soon for another class soon. Okay, take care everyone and stay happy during quarantine, please.